Hello, this is the exam 3 walkthrough, Alvin Gibbons, and I will be showing you how I worked through these problems. To do that, I'm going to share with you my screen so we can look at. All right, so for question 1, it asks us about the different human diseases that have been talked about, and it wants us to list out what how these terms are associated with the diseases. And so we got triploid, inversions, deletions, monosomies, duplications, aneuploidy, translocation, trisomy, and non-disjunction. So the first question, part A, is which of the above is associated with Patau syndrome? And that is a trisomy, aneuploid, non-disjunction. For part B, which of the terms is associated with a conventional form of Down syndrome, is trisomy, aneuploidy, non-disjunction. No, aneuploidy is just a different or an irregular count of we have chromosomes. Aneuploids would have 20, well, they'd have plus chromosomes. So 40, we have 46 total chromosomes. And so somebody with a trisomy or a monosomy would have plus 1 chromosome, and a monosomy would be a minus 1 chromosome. And so you'd either have 47 or 45, depending on that. And so for the trisomies, we know they have 47. Disjunction means that either in meiosis 1 or meiosis 2, meiosis 1 or mitosis, there was a chromosome that didn't split. It just kind of it stayed connected through the process. And so that's the cause of these, is non-disjunction. Turner syndrome is monosomy, again, aneuploid and non-disjunction. D is that, and that is a deletion. Okay, and then E, it's kind of a sneaky one, which is associated with type A, hemophilia, that is inversion. Part 1. Question 2 is asking us to examine the E. coli strand. So let me get some paper copy so you can see it a little better. The work that I did is I first recognized that transcription happens in the 3' to 5' direction. With 3' here, 5' here, and I found the primers. They fit into where they're both 3' on this side, 5' on this side. And you can match it up. You know, this bottom thing that I wrote in is a complementary strand, but in this case, because these are RNA to use, we know that these are built off the complementary strand. So the complementary strand to this RNA is in fact what's the word? It's not coding. Our template strand. Thank you. Okay, so that's A. Part B is what is synthesized, and so we just starting 3' end, we got a CGA. So you can see the G and the C and the T turn to CGA, and you just go this way. And because description goes from 3 to 5, we know that it's going to copy from this direction to this direction, which answers question C. No, just kidding. We know the order for question, or for part B. For question C, it's asking us which direction the fork is moving. Well, because we are told that this is the lagging strand right here, we know that the replication fork is going to go this way because the leading strand goes from 3 to 5, but in this case 3 is going to be on this side because how do I explain this better? Because this is the lagging strand, this side 5' of the lagging strand, its complementary strand, the leading strand, would be 3' on this end and 5' on this end. From 3 to 5, we know that it's going from right to left, right to left. And then with that said, which of the two primers is first synthesized? Well, if we're starting a replication fork here, it opens hypothetically to right here, and then primer B would come in first. And then it would open further, and then primer A, and then the polymerase would come in and attach to primer B and synthesize this way, and then it would leapfrog back to primer A and then synthesize this way, and that's what that's about. So that's part 2. Question 3 is a fun one, I think. It's super easy. It's asking us about paracentric inversion. The way we know that is because it says that suppose these two chromosomes, let's see, where is it? Okay, so there's a crossover between T and U. Okay, so T and U do not include the centromere, so we know that it's paracentric, it's outside. So if it were to be between the N and the O, yeah, the N and the O, or anything like that, then we know it is pericentric. And so then what are the 4 products? Well, the first one would be normal chromosome, but then as you're going down the line, once you get to the T, you jump down to where the U ends, and so you go TSX. That's what I did here. And so you see what I did? If there's a crossover between T and U, draw a line between T and U to tell you where that crossover is going to be. Jumps over to this side, and vice versa, and vice versa. And that's how we get these 4 products in for it. So for part 2, an inversion chromosome, whole chromosome end. And then in the third one would be here with the normal, and we go down, and as soon as we get to the T, we jump down to the U and go. So now we have 2 centromeres, which is a problem. But that's what it looks like. And then for part 4, kind of do the same thing as 3, but we start here at the X on the normal, work our way to the T. Once we get to the U, we jump down and then go back out. 4. Pretty easy, straightforward. The book is really helpful. But obviously this is the test, so because it's a fun process, I remembered it. Okay, so for part 4, this one is asking us about transcription. Okay, so which strand for lower is the template and which is the coding strand? So I said that the bottom is the coding and the top is a template strand. Again, we're going in the 3 to 5 prime. However, and we're going from left to right. And it says that right here. Okay, or from right to left, excuse me, right to left, right to left. So I know I wrote it on here as the template on top, coding on bottom. Doing the multiple choice test, I realized that these are indeed switched because the template strand is the usually is where it starts. So that's wrong. Ignore my work, but know that the template strand in this case goes on the bottom, coding strand on top, which actually probably means that I did question 2 wrong. I hope not, but we'll see. Okay, so moving on. B, what is the sequence produced? And so working our way this way, what I did is I just copied the template strand. In this case, it was the coding. What I did is the coding strand. I copied it all down, flipping out the T's for the U's. Done it in reverse, and so I should have done this side T's for the U's as I've transcribed it. And then in which direction is in which direction, left or right, along the coding strand relative to the +1 site would the -10 and -35 sequences be? Well, because it's going from right to left, we know that it had to start on the right. So it's going to be the right. And then the terminator, because again, it's going from right to left, we know that it's going to end to the left. So you do number 4. So that's it. Super easy. Have any questions? Feel free to ask the teacher, Sister Johns. But that's it for this week. Thanks.